Mirror life refers to hypothetical organisms with reversed chirality compared to natural life. Mirror life's molecules are mirror images of those found in Earth-based organisms. Advances in synthetic biology suggest creating mirror life forms may become feasible in the near future. Escape of mirror organisms from containment could cause significant ecological disruption. Mirror organisms' incompatible biochemistry allows them to outcompete native species for resources. Disruption of food chains by mirror organisms can lead to imbalances in the ecosystem. Mirror life poses a threat primarily due to its chiral inversion, rendering it unrecognizable to natural biological systems. Natural biological systems are adapted to interact with molecules of a specific chirality. Mirror organisms with reversed chirality could lead to unchecked growth and novel pathogens against which natural defenses are ineffective. DNA molecules exhibit a right-handed helical structure. The consistent chirality of DNA is crucial for precise interactions in genetic processes. Genetic processes such as replication and transcription depend on the right-handed helical structure of DNA. Proteins are constructed from left-handed amino acids, ensuring proper protein folding and function. Uniform chirality in amino acids enables enzymes to efficiently catalyze biochemical reactions. The exclusive use of left-handed amino acids avoids structural ambiguity and promotes stable protein conformations. Life based on mirror image molecules would fundamentally disrupt biological processes. Mismatched chirality prevents proper molecular recognition and interaction. Mirror image chirality would render essential functions like DNA replication and protein synthesis impossible. Scientists have successfully synthesized mirror versions of individual biomolecules. This includes creating mirror images of complex molecules like HIV-1 protease. This demonstrates the feasibility of manipulating molecular handedness in a lab environment. The creation of mirror life necessitates human intervention due to natural evolution's preference for specific chiral forms. Natural evolution results in a chirality lock-in, favoring one chiral form over its mirror image. Constructing mirror life is a deliberate engineering effort to reverse the chirality of biological systems. Mirror life organisms and their products exhibit increased durability due to their resistance to degradation by naturally occurring enzymes. This resistance makes mirror life organisms ideal for long-lasting medications and specialized applications. Mirror life bioreactors are resistant to contamination from standard biological agents, enhancing their reliability in industrial processes. The adaptive immune system employs chiral recognition to identify and bind to pathogens. Immune receptors are specifically shaped to interact with molecules of a particular chirality. This precise molecular fit is essential for triggering an effective immune response. Mirror pathogens possess molecules with reversed chirality compared to natural pathogens. Immune receptors cannot properly bind to mirror pathogen molecules due to chiral mismatch. The lack of binding prevents the immune system from recognizing the mirror pathogen as a threat. Mirror pathogens evade detection by the adaptive immune system, allowing unchecked proliferation within the host. Unchecked proliferation of mirror pathogens leads to chronic infections that resist conventional treatments. Broad-spectrum mirror pathogen infections can affect multiple species, posing a significant threat to public health and ecosystem stability. Conventional antibiotics target specific chiral structures present in natural bacteria. These antibiotics disrupt essential bacterial processes by binding to chiral targets. Antibiotics are ineffective against bacteria with opposite chirality due to improper binding. Mirror bacteria could potentially evolve to metabolize resources consumed by natural bacteria. This adaptation would provide mirror bacteria with a competitive advantage in specific environments. The ability to utilize the same resources allows mirror bacteria to thrive and expand their population. Mirror bacteria, if resistant to antibiotics and capable of using natural resources, can proliferate unchecked, Uncontrolled proliferation of mirror bacteria can disrupt existing ecosystems and outcompete natural bacteria. Ecological imbalances resulting from mirror bacteria proliferation highlight the need for research into new countermeasures. Mirror microorganisms lack natural predators or pathogens in standard ecosystems due to their unique biochemistry. The absence of natural controls allows mirror microorganisms to proliferate rapidly. Rapid proliferation of mirror microorganisms can outcompete native bacteria for essential resources and habitats. Mirror microorganisms produce mirror sugars, which are not metabolizable by most organisms. The introduction of mirror sugars into the environment disrupts the natural energy flow within the food chain. The inability to metabolize mirror sugars leads to nutritional deficiencies and destabilization of the food chain. Resource competition and food chain disruption can trigger a cascading ecological collapse. Declining native bacterial species and nutritional deficits and higher trophic levels contribute to ecosystem instability. Ecological cascades increase the risk of significant biodiversity loss and potential extinction events. Mirror life refers to organisms exhibiting reversed chirality compared to standard biological forms. Introduction of mirror life into an ecosystem could lead to rapid diversification due to novel biochemical pathways. Mirror life may experience rapid adaptation due to the absence of natural predators or competitors adapted to their unique structure. Mirror cyanobacteria perform photosynthesis using mirror image molecules. Mirror image molecules may allow cyanobacteria to utilize different wavelengths of light. Mirror cyanobacteria may resist degradation by existing enzymes, leading to unconstrained growth and altered energy capture. Unconstrained photosynthetic activity of mirror cyanobacteria could alter atmospheric cycles. Mirror cyanobacteria can disrupt nutritional cycles by incorporating and releasing mirror image nutrients. Changes in nutrient cycles can affect the food web and overall ecosystem health. Mirror life's unique molecular structure provides exceptional stability. This stability allows mirror life to resist degradation more effectively than natural biological entities. Enhanced stability makes mirror life a potentially long-lasting bioweapon. Mirror life's broad host range increases the risk of widespread outbreaks. Mirror life can infect a wide variety of organisms, including humans, animals, and plants. The ability of mirror life to infect diverse cell types poses a significant ecological disruption. Antimicrobial resistance complicates infection management by reducing the effectiveness of antibiotics and antiviral drugs. Antimicrobial resistance, whether natural or induced, poses a significant global threat. The potential for intentional or accidental release of resistant microbes could lead to widespread, untreatable epidemics. Mirror organisms are life forms constructed from molecules exhibiting the opposite chirality to those in natural organisms. Creating mirror organisms involves manipulating fundamental biological building blocks at the molecular level. Studying mirror organisms provides insights into the fundamental principles of biochemistry and the origins of life. Mirror organism research poses containment failure risks despite stringent safety protocols. Accidental release of mirror organisms into the environment remains a significant concern. Unforeseen ecological consequences may arise from the release of mirror organisms. Mirror organisms, once released, may evolve unpredictably in the environment. The unpredictable evolution of mirror organisms poses a significant challenge to risk assessment and management. Due to the inherent unpredictability of mirror organism evolution, a moratorium is suggested. Creating mirror bacteria is estimated to be feasible within 10-30 years if highly prioritized. Achieving mirror bacteria requires overcoming immense complexities. International collaboration and substantial financial investment, potentially reaching hundreds of millions of dollars, are necessary. Research into mirror molecules for therapeutic applications is ongoing and generally considered safe. Mirror molecules offer unique properties that can enhance drug efficacy and stability within biological systems. Enzyme-resistant drugs developed from mirror molecules can improve therapeutic outcomes. Self-replicating mirror organisms present a primary safety concern in biohazard labs. These organisms can proliferate and potentially disrupt existing ecosystems or biological processes. Unlike non-replicating mirror molecules, self-replication introduces significant potential risks. Historical lab leaks of natural pathogens demonstrate that even highly secure facilities are susceptible to breaches. These incidents highlight the critical need for robust containment protocols in laboratory settings. Continuous vigilance is essential to prevent future lab leaks and maintain the integrity of research environments. Mirror life could escape containment due to human error during experiments or maintenance. Equipment malfunctions can compromise safety barriers, leading to the release of mirror life. Deliberate, malicious actions could be aimed at releasing the artificial life form from containment. The escape of a mirror life form could result in significant ecological disruption. Unforeseen health risks are a potential consequence of a mirror life escape. Depending on the nature of the organism, broader existential threats could arise from a mirror life escape. Synthetic organisms are engineered to depend on synthetic nutrients not found in natural environments. This dependence on synthetic nutrients prevents the organisms from surviving and reproducing outside the laboratory. This nutrient dependence serves as a biocontainment strategy, limiting the spread of synthetic life forms. Mutations can arise within synthetic organisms' genomes over time. Some mutations enable organisms to utilize natural substrates in the environment. Adaptive evolution allows organisms to bypass dependence on synthetic nutrients, potentially leading to survival outside controlled conditions. Synthetic organisms can acquire genes from other organisms through horizontal gene transfer, HGT. Acquired genes encoding metabolic enzymes enable synthetic organisms to utilize new natural substrates. HGT-mediated adaptation represents a route for evolutionary escape, potentially compromising biocontainment strategies. Mirror life, once released, resists conventional eradication methods due to its unique biochemical structure. A simulation visualizes the rapid propagation of mirror life across diverse ecosystems. The simulation demonstrates mirror life's ability to quickly adapt and spread within various environments. Antibiotics are designed to target specific molecular structures in organisms. Mirror life possesses reversed chirality, preventing antibiotics from binding effectively. The reversed chirality of mirror life renders antibiotics ineffective due to their inability to disrupt cellular processes. The immune system identifies pathogens through their specific molecular structures. Mirror life's reversed chirality prevents immune recognition and response. The reversed chirality of mirror life results in a lack of natural defense mechanisms against its spread. The precautionary principle advises prioritizing preventative measures when facing potential irreversible harm, such as from creating mirror life. Funding restrictions on research that could lead to complete mirror organisms are a key preventative measure. Establishing international treaties is crucial for regulating activities related to mirror life creation globally. Public dialogue is essential to address the ethical implications of mirror life research. Scientists advocate for discussions involving policymakers, ethicists, and the public to weigh potential benefits against existential risks. Conferences are planned to facilitate these discussions and inform future policy decisions. Research into partial mirror systems, like ribosomes, provides an alternative approach that minimizes risks while maintaining potential benefits. Pharmaceutical production using partial mirror systems allows exploration of mirror life advantages without creating full mirror cells. Focusing on partial mirror systems reduces the likelihood of unintended consequences associated with full mirror cells. The escape of mirror life from a laboratory environment poses significant risks to human health. Exposure to mirror organisms can lead to chronic conditions and diseases difficult to treat with conventional medicine. The unique biological structure of mirror organisms could interact unpredictably with human physiology, resulting in unforeseen health complications. Introducing mirror life into natural ecosystems poses a high risk of ecological collapse. Mirror organisms can outcompete native species for resources, disrupting established food chains. Ecological imbalances caused by mirror life can lead to irreversible damage to biodiversity and ecosystem functions. Strict prohibitive measures are crucial for preventing the escape of mirror life. International cooperation is essential in mitigating the threat mirror organisms pose to global health and ecological stability. Collaborative efforts ensure that mirror organisms remain a theoretical concept, not a tangible threat.